Okay, our next speaker, Lynn MacDonald from CSIRO Land and Water in the Carbon and Nutrient Cycling Research Group based at Adelaide. And she's currently leading the South Australian component of the National Soil Carbon Research Program. Also quantifying carbon, nitrogen and phosphorus content of soil organic matter and researching biochar on soil biology. And she's going to be talking to us about soil sampling strategies for carbon.
Excellent, thank you. So yeah, just a little bit of an outline of what I'm going to go through today. We're going to take a little bit of background just in terms of what we're dealing with to sample. Then I'm going to run through a bunch of different strategies for sampling paddocks. So we're going to look at the 25-metre grid. We're going to look at paddock scale random sampling and paddock scale grid sampling, and also what we might do if we need to think about zone sampling of paddocks. I'm then going to finish up with a little introduction to the National Soil Carbon Research Programme with particular kind of focus and highlights from what's going on in South Australia. So just to start off with what we're dealing with, we know that soil forms a continuum in space. It's not a defined unit. We know that it has vertical dimensions, so we need to consider depth. We know that there are diagnostic horizons that are not visible from above ground. And we know that soil can be difficult to classify. We know that we're dealing with a variable landscape in terms of soil type and landscape features such as slope and undulation. We know that it changes in terms of texture and colour and mineral composition. And management history, of course, has a fair bit of a large impact on soil carbon and on variability. And I guess just trying to visualize this, I went into Google Maps and just took a little look at the satellite images and grabbed various shots from across the Australian landscape. And I guess here I just want to get across the point that we can visualize this variability in the landscape to a certain extent. And I guess maybe just highlight that out here in a snapshot of WA, we have some pretty big regular paddocks, and they are across a fairly flat kind of sand plain landscape. And that contrasts a fair bit with the type of remnant dune type systems that we get in a lot of other Australian agricultural systems. So we need to consider what we're sampling. Our aim is to collect a representative sample of whatever we have decided. But in order to define a representative sample, we need to carefully consider what our purpose is and what our spatial context is. So just taking a little bit of a closer look at purpose, are we looking to determine change in soil carbon content over time? Well, to me, that's not nearly specific enough. It's way too fuzzy around the edges and doesn't really define what we need to know in order to sample correctly. We need to define the spatial area that we're looking at, and we also need to look to see if there's any temporal requirements. So if we're looking at change over time, is there a particular sampling gap between sampling that we need in order to measure a minimal change in carbon content? So I guess looking to better define that question, we might expand the question, or the focus is to determine change in carbon content of a specific paddock after however many years of management change. And then we're beginning to get to the nitty-gritty in order to help us design our sampling strategies. So the sort of minimum requirements that we need are to collect a representative soil sample or samples. We're looking to sample to a minimum of 30 centimetres depth. Bill mentioned that we need an accurate estimate of bulk density and also an accurate measure of soil organic carbon content. We also need to think about the confidence or uncertainty associated with our results, and I guess that's why up here we need to think about the number of samples that we're required to take and how that influences our confidence in the results. So 30 centimetres depth, why? I guess we know that the majority of soil carbon is in the top 30 centimetres, and most of the management management changes impact that top 30 centimetres. There are, of course, always exceptions to the rule. So if we're specifically looking at engineering soils at depth, so clay spreading or delving type approaches, we might need to consider a depth greater than 30 centimetres. And particularly if you're changing to a crop species that has deeper rooting depth, then you might want to consider going beyond 30 centimetre depth because there'll be carbon inputs at the lower zone. But again, that is helping to define, or it's key to define the purpose and the context before defining the sampling strategy. And I guess the other point on this slide is that soil type has an important influence on soil carbon content. So here we've just got a bunch of common Australian soil types, and up above is the change in carbon content with depth. Moving on to bulk density, simply the weight of soil in a given volume. And we need that in order to calculate total organic carbon content on a unit area. So we know that bulk density generally increases with depth, and bulk densities above 1.6 can restrict root growth. But important to our carbon calculations, we need to multiply the depth of our sampling, multiplied by the bulk density, and multiplied by the carbon content. So if we have a 30 centimetre depth and a bulk density of 1 and a carbon content of 1, then we have 30 tonnes of carbon per hectare. The complication with bulk density comes when we step into gravel soils, and we'll need to apply a correction factor to the calculation in order to account for the gravel content. And sampling soils greater than 10% gravel becomes difficult at more than about 10%. And I think there's one or two people in the audience that have particular experience with sampling bulk densities in difficult soils. So, moving on to our sampling strategies, I think most people will probably be or have heard of the 25-metre grid sampling approach, where a 25-metre grid at 5-metre intersects is put down within a paddock, and it can be laid out relatively quickly. It's a common approach, and it tends to minimise the influence of landscape variability on the results. And this is the approach that's been taken in the National Soil Carbon Research Programme. So just to run through that, we've selected a random GPS location, which defines the bottom corner of the 25-metre grid sampling. We've got 5-metre intersects, so 36 possible sampling points across that grid. At 3 of those, we take a bulk density measurement at 3 depths, so the 0 to 10, 10 to 20, 20 to 30. And we create a composite sample across that from a collection of 10 samples, which are bulked at the 3 different depths again. So that's the approach that's been taken within the National Carbon Project. The advantages of this grid type approach is that we can go back and revisit the precise locations. It's good for monitoring purposes, so we can go back to that landscape. We've minimised the landscape variability, and it provides a good data set where we have high replication or high representation. So for example, in the South Australian National Project, our aim is to determine the impact of management in red brown earth soil types on the carbon content. So we have over 200 sites on red brown earth soil types, so our representation is high. We have a stratified design, so we're specifically sticking to red brown earth soil types. The small size of the sampling grid ensures that we don't cross too many soil boundaries, and we've got this soil times land use combination, which is replicated. However, if we're thinking about sampling at a paddock scale, and our specific purpose is to define the carbon content of a specific paddock, then we want to encompass that paddock scale variability. And if we plop down the 25-metre grid sample within a great big paddock, we're actually losing that paddock variability. So we might like to think about an alternative strategy to get a better sampling or better estimate of the carbon content within that paddock. So an alternative approach would be to randomly scatter a number of sampling points across the paddock and collect samples from there. If you're looking to gain more information about the variability within that paddock, you might like to keep those samples separate. But if you're looking to form an average carbon content, then you might like to composite the sample across those sampling points. I guess when you go out into a large landscape, it can be a little difficult to randomly allocate sampling points and to be happy with your random allocation. You might by chance get clustering in particular areas. So to avoid that, you could put over a larger sort of grid scale or grid across the paddock, and then select the specific grid intersects that you want to sample across. And the advantage of that would be it's easier to keep track of where you have sampled and to distribute your, the number of samples across the paddock in a more uniform manner. So I guess the example that's up there is an image that's grabbed from the kind of sand plains in WA. And again, we've got this probably fairly uniform paddock. And I guess that in some ways is fine, but if we have any sort of underlying variability in our soil across that paddock, then we might run into problems. So just to kind of highlight, if the blue zone represents a different soil type, so if we've got contrasting soil types within that paddock, and we've randomly thrown out our points of collection for our sampling, it might happen that we only have a couple of those samples falling within one soil type, and then we run into potential issues of misrepresenting the carbon content of that paddock, particularly if you go back in subsequent years and your random sampling falls out at different points. You might, on that return trip, find that you have more sampling within the alternative soil type as opposed to your first sampling. So the confidence of your results in that case could be lessened. So if there is knowledge about underlying variability in your paddock, you might like to take that into account when you're designing your sampling strategy. So it might be appropriate to redistribute some of those sampling points so that you have better representation of the two different soil types on an area basis. And I guess just a second example there on remnant dune type systems. It's very obvious in this half of the picture that the hills of that dune system are managed quite differently. And that if you're defining your sampling based on a management strategy, then obviously that shrubby type vegetation on the top would be inconsistent with the rest of the paddock and therefore wouldn't be sampled. It's less obvious on this side of the paddock, where you've still obviously the dune system going on, and you have variability in your landscape characteristics and potentially your carbon content. And it might be important to take that type of variability into account when you're designing your sampling strategy. And in that specific case, you might like to take a similar approach to the underlying variability in soil type and actually provide a zone stratification type approach to ensure that you're incorporating both those hill slopes and swales in the sampling strategy. So in summary, quickly, the minimum requirements are to provide a representative soil sample to a minimum of 30 centimetre depth, and will require an accurate bulk density. In order to look at confidence or uncertainty, we'll also need a number of different replicates samples in order to calculate that. It's important to carefully consider the purpose. So is the purpose of the soil sampling to look at change over time? Is it to compare two different sites, or is it to improve particular understanding of variability? Again, it's important to consider variability. So what background information is known that can be used to inform information on variability? And if there is a change in management practice, will variability change with that management through the two different sampling periods? And I guess I just wanted to highlight that the ISO standards for soil sampling for carbon accounting purposes emphasize that sampling strategy is driven by purpose, but and that the general rules that are applied have to be adapted to that specific purpose in the specific situation. So it is a case of kind of adapting general guidelines, and there isn't specifically one sampling approach that's going to suit all situations. So just to highlight some of the SCARP work that's going on within the national project. The larger objectives of the project are to define a nationally consistent method for quantifying soil carbon. This phase of the project had a lot of discussion before we got to the sampling design across the project. And then beyond that, we're looking to identify in regionally specific areas land management strategies with the potential to build soil carbon. And then, as John's talked about before, there's the development of cost-effective methods for measuring or quantifying carbon stocks. So one component of that is the MIR predictive approach, and the other is the use of automated devices for bulk density, which might make life easier in some cases. The data, of course, is flowing into the national accounting system to improve modelling systems there. So the sampling approach we've run through already, and it's almost consistent across the programme. However, there are always exceptions, and I guess that just highlights the need to adapt the methodology to specific context. So in the rangelands projects, where they're dealing with very large spatial scales and very large paddocks, their grid sampling is a lot larger than 25 metres. I think they're working with a 100-metre grid sample. And Diane Allen's written a nice review of sampling designs specific to the Greys context. So in South Australia, what have we been up to? We are aiming to identify management practices with the potential to change soil carbon content. We've got a relatively small project, so we've only got 200 sites, and we decided to focus on red-brown earth soil types. And we have two target areas, the Mid-North and the Eyre Peninsula, and they are fully sampled at the moment. We decided to break down any sort of artificial boundaries around our kind of land management systems. So we've included everything from permanent pasture to continuous cropping and all the sort of mixed rotational systems in between. So we're treating our sampling set in more of a regression approach, where we're looking at the intensity of agriculture within the system. We've taken a random sampling approach. We have no knowledge of management before we go out there, so we're very reliant on the landowners providing or returning the land management questionnaire. And I know that I'm going to have a pretty unbalanced statistical design. So within the project, we are going to have three major data sets. We're going to have the management histories, which I'll talk a little bit about. We are pulling climatic and topographic data based on the GPS coordinates from the silo data type databases. And we'll have our soil organic carbon content and the fractions that John's talked about before, so particulate organic carbon, humus, and the resistant components. And in order to understand what's influencing soil carbon content, we'll be looking at correlating the management histories and the topographic data to those carbon contents. We know that at least that soil carbon content is influenced over many years, so we've collected the management data. And the type of information that we've collected, oh dear, is, can I remember, a bunch of information on crop type, tillage management, stubble management, and various aspects of pasture management, so grazing aspects and the likes. And that database is fairly large, and it has provided one or two headaches, but we have got there, and it's a very valuable data set in order to understand our soil carbon. It also fits into probably filling some of the gaps that Bill was talking about in terms of need to improve our understanding of what's going on there in the landscape. So this is nice, up-to-date information, hopefully. And the type of things that we can do with that data is look to see, because we've gone out there in a random manner into our landscape, we can make comment on the proportion of crop systems or mixed systems within our red-brown earth soil types in the target regions. We can look at the sort of frequency of incorporation of legume rotations within the management data in the last 10 years, or the frequency or adoption of no-till type practices. And then we can define our questions in terms of, we might like to ask how many pasture years are required to significantly alter soil carbon content. And that final little schematic is a look at the tillage management or trends in tillage management over the last 10 years. And we can see the adoption of no-till and reduction in conventional till and minimal till across the region. So just where we're at with this at the moment, our field sampling campaigns are complete. We have 130 sampled in the mid-north and 70 sampled on the Eyre Peninsula, 70 sites that is. We've got over 900 samples which are going through various pipelines. In terms of the carbon analysis, our MIR spectra we have across the whole data set. Our fractionation is a good way underway, and that will be used to update the prediction algorithm to get a red-brown earth specific prediction algorithm for these different fractions. One big headache within the project has been our carbonate soils, because 41% of those samples are carbonate, and in terms of analysing samples for carbon, that requires a bunch of pre-treatment with acid in order to get rid of the carbonate and measure the organic carbon content. So that's been a lot more time consuming than we thought. And I guess that just highlights the data sets that we're going to have in terms of the management, the environmental, and the carbon data sets. So from there, we're looking to understand the variability in our sample population, first of all purely with the management questionnaire, and secondly looking to identify management drivers in soil organic carbon content over these two regions. In terms of looking at uncertainty, we do have a bunch of spatial sampling. So we've got some samples where we have the grid, the 25-metre grid, and instead of compositing the samples, we have the 10 samples kept separate, and we also have Paddock scale sampling at those sites, where we've scattered 10 sites randomly across the paddock, and we're looking to assess the uncertainty associated with predicting measuring the carbon content in these paddocks based on the 25-metre grid and the paddock scale sampling. I guess just to finish, across I think this was largely the mid-north, this is carbon contents from last year's data. We're looking at carbon content varying from 22 to 64 tonnes of carbon per hectare. And the next stages of our course are to update that with the fractions, to look at the distribution of the organic matter fractions, and also to correlate that data to the management and environmental data to look at what's driving soil carbon content. So just some acknowledgements, firstly to the participating South Australian farmers, because they have been very good at getting involved and getting back the management information. We've had a sampling crew of over 10 strong out there, really putting in the hard work. And the lab crew have been pretty much up to their eyeballs and working pretty hard over the last while. So, thank you.
Lynn, very interesting talk. Can you tell me, I'm interested in your estimates of bulk density, because if they're done using a drilling rig with a coring device, my experience in calibrating neutron probes is that it's very, very difficult to get a reliable estimate of bulk density from a core sample, from a simple drilling rig. Can you comment on how you...
So South Australia, for the bulk densities, we're actually hand coring.
Oh, good.
Yeah. I think, we recognise that there's likely to be compaction with the drill rig. Yeah. And our soils aren't actually within the part of the project that's looking at the bulk density, the rapid bulk density measures. So Dan Murphy in WA's done a fair bit of work with that on the sands out there. And I think there's been other work. Update me, John. Yes. But I'm not sure what region he's been concentrating in or soil types. So, but yeah.
So it's really important to get a good estimate of the density.
Yeah, and it's one of the parts of the sampling that takes the longest. So it'll take as long as getting the composite sample together. So.
Lynn, just a sort of a statistical view, how big is the difference that you want to estimate in changes in soil carbon? Because the accounting seems to take it down to tonnes per a 1 tonne difference. And what probability do you want to have in detecting that difference? Because that really determines your sampling number.
Yeah, that is going to determine sampling number completely.
So
I wouldn't
is it an impossible task to get to a soil
1
assessment
tonne
based on a carbon accounting number?
I think the carbon accounting will have to define what the minimum limit of change is, and that will help define how many samples need to come out the ground in order to provide the type of robust data. Yeah. But at this stage, I would not like to take a gamble on what might be our minimal detection limit.
You mentioned that in order to get an assessment and an appreciation of the uncertainty, that you could analyse the 10 cores individually and see the spread of results. Did you also consider going back to some of the other sites of the 36 pattern grid and take another 10 samples and just look at the variability using that approach?
Sorry, which 36 sites?
When you
the
drew
other
the
grid.
5x5 grid and you took 10, I think I understood that you took 10 of those 36 sites. Did you ever also go back and take another 10 different grid points?
No, we haven't done that, but that would be interesting. Yeah.
So just following on from both of those questions, in respect to the last one, are you bulking those 10 samples before you do the analysis, or are you doing 10 separate analyses and then calculating a mean?
That'll be done on 10 separate. They've been deliberately kept separate.
So in that case, Roger, your question, you would expect that you wouldn't get a difference because you'll get a robust estimate of the mean. But related to the previous question, I think if I understood Bill Slattery correctly, he said that the total error in the soil measurement was of the order of 9 to 22 tonnes per hectare under the strategies that he had available to him. What are you expecting your error to be, and what do you think an acceptable target is?
Ooh. Yeah, I am not sure at this stage. I think when you talk about, you know, errors of 22 tonnes and you look at my carbon distribution across that area that starts at 22 tonnes and goes up to 63, for accounting purposes, I think it's going to be quite different to what I'm trying to do within the carbon project. I guess, yeah, I'm going to have an awful lot of uncertainty associated with my management data in order to try and understand what's going on as well. So there's another level there as well.
It seems to me it's important to get on top of this issue because it may well be that you can end up taking a lot of samples that don't actually give you any additional information in terms of error, because even under the strategy that you're using, you're not taking anything like enough samples to characterise the spatial variability. So there's a benefit cost issue that has to be addressed here in terms of the additional value that accrues from those additional samples, and I'm not all that persuaded so far that benefits necessarily accrue.
John did some work on some of his perennial sites where he looked at how many samples he would need in order to account for the variability within some of the perennial paddocks. But again, that is going to be very much dependent on the type of landscape that you're in and the amount of variability that you're dealing with. Which is pretty big issue to tackle, and it needs to be done.
Seems like we're getting a few questions, but I've got time for one more. So.
Mine's part of a comment actually, which relates to what Rob said and also what you've just been discussing in relation to sampling. And there's two issues. One is obviously the science of what we're doing and being able to be accurate and be able to predict. The other issue is in relation to carbon accounting and the cost of sampling. Mm-hmm. And to actually be accurate enough, it's going to cost you more than the carbon's worth half the time. So the question about how much sampling and how accurate will actually depend to a certain extent on the value of the carbon. So that really needs to be factored in terms of when you start talking about certainty and sampling strategies. Well, who's paying, and that'll be determined by carbon price.
Mm. Thank you.
Speaker is Ron Smernik from University of Adelaide. He's a senior lecturer in the School of Agriculture, Food and Wine, and he's been involved in soil organic carbon research over the past 14 years since starting as a postdoc in 1997.
Thanks, Matthew. Right, I'm actually quite familiar with this lecture theatre. This is where I give most of my lectures. Good to see a few faces who actually have attended those lectures. Hopefully they're not the disruptive ones. Okay, a little bit for those who don't know who I am, about me, some stuff about where I was born and where I've lived and what I did as education. My wife seems to think I overdo the country boy thing. As you can see, I was born in Griffith. My parents were, at the time, share farming my grandparents' farm. I like to think I was really involved, but my wife points out that I probably couldn't hold my head up by the time I moved to Sydney. So I'm actually a bit of a city slicker. And also, in terms of agriculture, I came to it reasonably late in my career. My background is actually in chemistry and straight science, and it was only from my postdoc on that I've been involved here. But that is starting to be a fairly long time. So in summary, I'm a scientist, so my training is in chemistry, and my research experience is in soil carbon generally. Here, I worked initially with Malcolm Oades, and since then kind of independently, but a lot with Jeff's group in CSIRO. Okay, second thing, what is biochar? And in terms of material science, it's a pretty straightforward question, and really it's just charcoal. I think most people know what charcoal is, but in case you don't, there's a bit. You can pass that around. As far as me as a chemist goes, it's just another name for charcoal. But in this context, in the soil carbon context, it has a bit more of a specific meaning. It's charcoal that's produced with the purpose of adding it to the soil. It's produced under conditions of pyrolysis of biomass rather than combustion of biomass, inefficient combustion of biomass. I'll go into that a little bit in a minute. And that biomass is generally waste biomass, either forestry waste, urban green waste, rice husks. There's a number of different types of waste biomass that can be utilised, and the other way is to specifically produce biomass crops and turn them into biochar. Heating temperatures we'll see in a minute that is quite important. Generally, the heating temperature for the biochar is around 400 to 600, but there's quite a bit of variability in how people are producing this stuff. Okay, that's what biochar is. The biochar concept has these three prongs. The first is producing energy from the biomass. So by pyrolysing, by heating biomass in the absence of oxygen, it's exothermic, you produce energy from that, and it's one of the many ways that you can get energy from biomass. The second is that it has potential for storing carbon in soils, and the third is the potential for improving soil productivity. And I'll go into more detail of each of those as we go through. Okay, the first thing is, first part of it, actual pyrolysis, producing energy from biomass is the bit I know the least about, so I'm just parroting what I've heard from other people in the area. So basically what we're doing is heating biomass to around that 400 to 600 degrees Celsius without air or oxygen, and when you do that, you convert about 50% of the carbon into volatiles, into some CO2, but also gases and oils, which you can then combust with oxygen to produce energy. So that's sort of the energy component of it. You get some residual heat, which you can use to sustain that pyrolysis. So it's self-sustaining. You're not putting energy in to heat it up. Obviously, when you're starting the process up, you'll need to have a source of energy, but overall, the process of pyrolysis is self-sustaining, and you get a net energy output, and that's the energy side of it. There's some advantages in pyrolysis as a means for getting, if you like, bioenergy or energy from biomass. It's relatively low tech. It's efficient at fairly small scales, and this is important when you're talking about waste biomass, which is bulky and not particularly valuable, that if you can produce the energy on a small scale, you reduce transport costs. You can use most biomasses as a feedstock, not particularly picky, and the benefit in terms of greenhouse gases is that it's a replacement for fossil fuel, so we're getting energy directly from that biomass. And compared to other technologies for getting bioenergy, it's and efficient. I've got that in inverted commas because there's a lot of ways to define efficiency in these processes, but you're certainly converting a greater proportion of that carbon into something that you're using than if you're producing ethanol from sugar or corn or biomass, where you've got a fermentation procedure going on, and a lot of that energy that's stored in the biomass is actually being utilised by the bugs that are doing the transformation. Okay, the second part of it, which is probably the one you guys are most interested in, is storing carbon in soils. And the question is why we want to do this. I've given similar talks to grower groups and so on. And the answer, I go into a little bit more detail, but basically we're talking about greenhouse effect and global warming and all that kind of stuff. And I'm assuming I've assumed that other people have covered this already, or that we're already over the idea of why we'd want to store carbon in soils. Going into just a couple of specifics of storing carbon in soils. Why are we targeting soils? Well, they store a lot of carbon, more carbon than there is in vegetation and more than there is in the atmosphere, or actually more than the combination of those two. And the second part of it is that the soil carbon pool, these terrestrial pools, as compared to geological pools, which are really big, or oceanic pools, which are bigger than the terrestrial pools, the thing about the terrestrial pools is they're very strongly coupled to atmospheric CO2. About 16% of the CO2 in the atmosphere is pulled down out of the atmosphere by photosynthesis each year. But of course, nearly all of that is subsequently released through respiration by the plants themselves, by animals that eat those plants, by insects, bacteria, fungi, and so on. About 50% of that respiration happens above the ground, and about 50% of that respiration happens in the soil. So in the case of biochar, we already have the process by which the carbon is being taken out of the atmosphere. What happens to most plant material after it dies is it's broken down relatively quickly, and John has talked quite a bit about this in his talk. He also mentioned that charcoal, by contrast, is relatively inert or stable. It doesn't get broken down quickly by bacteria and fungi. And the idea of biochar, which if you do a Google image search for biochar on the web, you always come up with this particular diagram. What it's showing is that generally what's happening in the absence of utilising biochar, what we're doing by photosynthesis is we are fixing a lot of the carbon dioxide, but respiration is balancing that, respiration both by the plant and indirectly through the soil. The length of time that carbon that's been fixed by photosynthesis that is staying out of the atmosphere is fairly short, and we get released relatively quickly straight back to the atmosphere. The idea with biochar is to interrupt this process. So we're taking that 50%, say, of fixed carbon that isn't directly respired, that comes through in plant residues, pyrolysing half of that 50%, so 25% of the photosynthesised carbon is directly converted into carbon dioxide. We're getting some energy benefit out of that, but the remaining 50%, so 25% of total photosynthate, is then converted into a stable form. So by doing this, by converting 50% of the biomass to biochar, we're effectively shunting that 50% out of the rapidly cycling carbon pool. So key to the whole concept is this idea that biochar, is stable in soil, and first couple of speakers talked about this. This is something that's generally accepted, and there's sort of three reasons why we're pretty confident, or very confident, this is true. We've measured the rate directly of biochar carbon